First, let me say I'd like to thank the organizers for the opportunity to give this talk today, for organizing this fantastic meeting. I'd also like to thank them for the support to bring my students here. They're going to benefit greatly from the opportunity to see all of these just fantastic talks and being able to interact so directly with so many great people. Now, I'm Andy Spakowitz. I am in chemical engineering and materials science, so I'm an engineer by training. Although I would say that we have a very interdisciplinary research group in that I've graduated students from 8 different departments, and so I really benefit a great deal from having ideas coming from various directions, and I welcome your ideas as well in this talk. So what I'm going to talk about today is a set of theoretical, computational, and some experimental work that we've done to understand epigenetic regulation, but generally, chromosomal organization and dynamics, okay? And just to get us on the same page, let me show you this picture of a nucleus stained for DNA. And as you can see, it's highly heterogeneous. And there are regions which are much darker and regions that are much lighter. The darker regions are called heterochromatin. The light regions are euchromatin. Loosely speaking, the heterochromatic regions are the ones which are transcriptionally silent, whereas the open euchromatin is open for business, and it's expressing that genetic information. Okay. Now, a couple of points that I would make is that when you look at this, you notice that the dark and light does indicate DNA content, and so there is some aspect of it where there's a differential amount of DNA. There's a density difference between the two. You'll also very easily notice that there are pockets on the interior, but then lining the exterior of the nucleus, those dark regions do tend to localize, and that's going to be important to something I'm going to talk about in just a second. Okay, so I don't think I need to convince you all that epigenetics is extremely important. It is the basis on which multicellular organisms can differentiate a cell, a stem cell to all of the different cell types in the body. So it's an extremely important problem in terms of our function. It's extremely important for a variety of diseases and for practical applications. Okay. Now, our long-term goal is for us to use develop some physical understanding of how this phenomenon occurs and what impact the physics has on the biological function. Okay, and so to jump right in, I think that to give you a sense of what I mean by bottom-up modeling, the approach that we're going to take is to sort of begin to develop our perspective by adding ingredients sort of systematically, in the sense that we aren't going to approach it by trying to throw everything at it at one time. Instead, I'm going to sort of try to cook a really complex meal just by adding one ingredient at a time. Okay, so let me give you the first ingredient that I'm interested in. So, of course, there are many epigenetic modifications that are relevant to regulation. A very important one happens to be histone methylation, and the particular methylation that we're going to be really interested in for this talk is H3K9, the methylation of the 9th position on the H3 core histone octamer. Okay? And so, core protein of the histone octamer, excuse me. So, so let me say a few words about that. This is the nucleosome core particle. It's sort of the fundamental unit of packaged chromatin. Okay, it's about 147 base pairs of DNA wrapped around a protein complex, and it looks like this. It's this beautiful structure which leads to some level of compaction of the chromatin. Okay, and then what you'll also notice is that in this crystal structure, there are these tails hanging off, and this happens to be the tail of the H3 core protein. And this little residue right there is the 9th position lysine, which can be mono, di, and trimethylated. Okay. Now, it turns out there's a very important protein called heterochromatin protein 1. In fact, there's not just one. There's alpha, beta, gamma. But for now, let's just say we're interested in HP1 alpha. Okay. And this guy has 2 domains. A chromodomain, which interacts favorably with this tail, this H3 tail. And then there's another domain, the chromoshadow domain, which is shown to oligomerize in solution. Okay, so you can kind of imagine that if you had a string of these nucleosomes and you had a region which was highly methylated, that would sort of bind preferentially this HP1 protein, and then the other dangly bit, the chromoshadow domain, would act as a glue that could glue this together and cause condensation into what would be ostensibly heterochromatin. Okay, so this is an extremely important protein for understanding epigenetic regulation, and so let's just build the basic model for how that binding would occur to a single nucleosome. And here we are aided a great deal by experiments that were performed in Geeta Narlakar's lab at UCSF, okay, where what they did was they took mononucleosomes, okay, where the tails were either unmethylated or methylated, and then they looked at binding curves for the HP1 protein to bind to those, okay? And what's really nice about a systematic biochemical study like this, and let me show you what the data looks like, is that it allows us to be able to put some numbers on what is the binding affinity for the HP1 to a unmethylated tail versus a methylated tail, as well as what level of cooperativity there is between those chromoshadow domains that are interacting favorably. Okay, and so the experiments are shown in the dots. It turns out that they did this not for HP1, they did it for SWE6, which is a yeast analog, but that is the data that we have at our disposal. So for our purposes, let's just say that this is good enough for nailing down the basic picture of what a model would be. And what they found is that for unmethylated nucleosomes, the binding curve looks like this, whereas for trimethylated, it looks like this. You can see there is a higher binding affinity towards the trimethylated, but interestingly, it still binds quite prevalently to unmethylated, okay? And when you extract the numbers for what the interactions are, it turns out that the binding energy between binding to methylated versus unmethylated is only different by about 1.5 kT. And the strongest component of it is the cooperativity, which is actually on the order of 4 kT. And so this is actually, I mean, I don't know if you're alarmist about this, but it turns out that all of the segregation of your chromosomes into heterochromatin and euchromatin is dangling just on a difference of 1.5 kT between the methylated and the unmethylated. Okay. Now, so that's the picture for our nucleosome for the binding of the HP1. So now what we really want to do is we want to be able to build up a model where now we have nucleosomes that are in an array which to our best ability can capture the physical behavior of chromatin. Okay, and of course, we ourselves and many others have done a great deal of work to understand how chromatin actually, the local structure and the properties of it. And in fact, there's quite a great deal of controversy with regard to whether it is a fiber-like structure, as in a 30 nanometer type structure, whereas more recent imaging of within living cells looks like it looks a little more like a beads on a string. And so what I'd like to do is I'm not going to talk about some of our work on the local nucleosome array stuff, but let me just say that my student Bruno Beltran will present a poster where he has looked at what is the effect of there being even a modest degree of heterogeneity in the linker length of the DNA between nucleosomes. And what he's found, and I'm going to let him explain why and what we've found. What we've found largely is that the properties of that array of nucleosomes is dramatically influenced by this slight degree of heterogeneity as far as where the nucleosomes are positioned. And I'm going to give you one example of what the biological implications of that may be in just a second. Okay, so I've explained that we have a basic picture for how nucleosomes bind HP1 depending upon their methylation. And of course, if we want to actually model a chromosome, right, we need to be able to extract what is the in vivo markings of the methyl marks on the chromosome. And here, if you take ChIP-seq data, in principle, you can just sort of read off this, and by developing a probabilistic description for what is the likelihood of each of these sites being methylated, we then can sort of read out and assign the nucleosomes as being methylated and unmethylated. Okay, so to a first approximation, this is sort of the basic ingredients for how you would talk about segregation into heterochromatin and euchromatin. So let me pause for a second and just admit that I'm a physicist, a polymer physicist. And so let me say a few words about what we expect to happen when you have an AB type polymer, meaning methylated, unmethylated type polymer, where one of the segments really loves to be next to itself and is indifferent or dislikes the other. Okay? So there's a long history of technological materials that are based on what are called copolymers. And it turns out that if you have a polymer with a segment of A and a segment of B, and even if they have a very weak interaction between each other, right? We heard earlier today about how interactions can be additive, very weak on an individual basis, but when you add many of them together, big things can happen, like phase segregation. And that is definitely the case when it comes to polymers which are connected together in a string. If you look at an AB copolymer, and let me be clear, this is the simplest possible copolymer you could imagine. If you crank up the interactions, which are described by a two-body interaction parameter, which is called chi for historical purposes, if you increase it, there is a transition between it being mixed to it being, there's an order-disorder transition. And what you'll notice is that the transition from here to here occurs not just at a threshold of chi, but it's actually chi times n, the molecular weight. And this is actually really important for the following reason. This is something that alludes immediately to the biology. How do you interpret a ChIP-seq sequence? If you see one gene in isolation, which is methylated, is it necessarily segregated, or is it weakly segregated, or does it remain in the euchromatin? What is the threshold for that? And it turns out from basic polymer arguments, there is a threshold which is not just the strength of the interaction, but also the molecular weight of the region which would cause it to segregate. Okay. Now, and we've done a lot of work on the basic physics of how these things segregate. But let me just pause for a second and also mention that there's very beautiful work that has been done in describing heterochromatin, euchromatin segregation. And in fact, the approaches that have been taken really complement a lot of the things that we do, in the sense that there's been this really remarkable work which has incorporated significant detail on a number of different epigenetic modifications that would drive the segregation. And it's highly, highly capable of drawing conclusions and relating to the experimental data. Okay? And I'm not going to talk about the details of these. I think we're going to hear from Jose tomorrow, I believe. And so certainly, these are really important. And I would contrast what we're doing in the sense that we are taking the ingredients and we're just looking at, okay, what's the result? We're just trying to see what happens. And so we are going to pale in comparison in terms of the success in describing experiments. But what I would also argue is that we can make certain inferences about basic physical phenomenon and mechanisms that are unbiased by the experimental data. And that's sort of what I'm going to emphasize today. Okay. So, all right. So, basically what we've done is we have all the ingredients. We now just have to simulate it. I don't want to go through the technical aspects of this. I didn't give you guys a workshop yesterday, but of course I am happy to sit down and talk about all of the aspects of how we model these things. The only point that I will make is that we want to be able to simulate chromosomes, full chromosomes and multiple chromosomes, even hopefully someday whole genomes of polymer. But we want to do it at a nucleosome level, and so it turns out that we have hijacked certain techniques from polymer field theory in order to make the calculations extremely efficient. Okay? And so I'm not going to dwell on this point right now, but just suffice it to say that if you are interested in doing these sorts of simulations, we are happy to talk with you about the details, and we're happy to, and we provide our code in order to enable that. All right. So first thing, let me just say this, is that in some regards, what I would say is that HP1, you could say, from our modeling perspective, acts sort of as a global regulator of segregation between heterochromatin and euchromatin, in the sense that if in our simulation we just simply crank up the HP1, there's a threshold on which this guy goes from being a mixed state to a condensed state, where there's a clear core and an outside peripheral region, which ostensibly you would say is heterochromatin and euchromatin. If you keep cranking up HP1, it actually over-condenses, and you begin to draw in the peripheral regions into that core. Okay, so that's just the basic picture of the physics, and I think that provides some statement about what essential role that HP1 could be playing in terms of modulating the degree of segregation in which genes are silent and open. Now, here's the other thing, and I alluded to this earlier. Remember I said that the degree of segregation is sensitive not just to the strength of the interactions, but also the molecular weight of those domains, okay? And so when you interpret a ChIP-seq sequence, it would be highly advantageous for us to be able to understand to what degree we can do a window average as you read along the ChIP-seq sequence and be able to say truly how condensed is a particular site, okay? So what we do here is we are providing the correlation between the degree of methylation as it is averaged over a window of a particular size. So this is the size of the window. And the correlation between how condensed something is as it's defined by the number of near neighbors and the degree of methylation. And what you find is that if you do not window average and you just base it on the sequence alone, right, you have many segments which are methylated but are in euchromatin. And there are many segments that are methylated methylated in euchromatin versus unmethylated and in heterochromatin. So you get false positives and false negatives galore. Right? Lots of them. But if you actually crank up the window average and you ask what is the optimal for classification, it happens at about 100 nucleosomes where there's a peak there. And what you can see is that if I color this chromosome simulation, where on the left we are just coloring it based on what is the methylation of each of the marks, versus the right where we actually do the window average, you can see the highly unmethylated regions as its window averaged are sitting in that euchromatin region versus the moderately versus the highly are sort of sequentially going like that. And I think the point to this is like this is actually in some regards I think useful. Whether or not quantitatively this is exactly right, the point that I would make is that as you begin to start to ask yourself what is not only how condensed something is, but also what is the tolerance for taking a gene and moving it from a region which is highly methylated and moving it to somewhere else where it may be methylated still, but its neighbors aren't. You have to ask yourself, what is the susceptibility for opening that up to the nuclear environment for being open for business, okay? And I think that that's where having this sort of physical picture is useful. Okay, so let me just say, I already mentioned this. Erez, who is one of the originators of Hi-C and gave a beautiful talk about contact maps. Hopefully, I think you're familiar with what this guy looks like. And so let me quickly just say that if you take the experimental data and you compare it to our simulations, what I'm going to argue is that it's okay. It does some things okay, right? Like you got compartmentalization, the basic picture, it's got some of that. It does other things, not so great, okay? So for example, there's a lot more enrichment right along the diagonal, whereas ours shows none of that. To be clear, this is a model where we have no loop extrusion factors. We have improved upon that in recent works, okay? But this is, again, just like a zeroth model where we just take the basic ingredients and feed it in. And so what I would argue is that given that the basic picture for compartmentalization arises just from one mark that is characterized by biochemistry, the interactions are ones that we just extract from Geeta Narlikar's lab and we just put it in the box and say, what does it do? We're okay with this, all right? And so I'm going to, how much time do I have? I probably just have 5 minutes. Okay, great. So then very briefly, let me just go through some points then. Is that if we're satisfied the basic picture has some relevance, okay, let's ask a few questions, okay? So first of all, you notice that in those other simulations that the heterochromatin is centrally located in that confinement, right? But we know that in cells for many cell types, not all cell types, but many cell types, it is peripherally localized, okay? And it's really not well understood what is localizing heterochromatin to the periphery. There are many proposed mechanisms for this to occur, and clearly there's a great deal of correlation between what are the peripherally localized ones and certain epigenetic marks and states, okay? But here's the thing that we want to do. In the spirit of us just asking the basic question of what do just some simple interactions, what would arise? Let me give you the simplest possible one. we take our model, and then we put in a non-specific interaction between all nucleosomes, not heterochromatin or methylated nucleosomes, but all nucleosomes and the periphery, okay? And then what happens? And the answer is that actually that alone very reliably will lead to heterochromatin being on the nuclear periphery as it's defined in the simulations. Now, given the shortness of time, what I'm going to do is I'm going to just say that Quinn is here, and he will give a poster, and I would very much like to encourage you to talk with him about the details of this issue of peripheral localization of heterochromatin. And so let me move forward. Let me say a couple of brief words about another thing that we're extremely interested in, and that is, every single cell cycle, you have to reestablish the methyl marks on the chromosome in the daughter cells, okay? And again, the mechanisms by which this happens is not well understood, okay? And on the one hand, you could imagine that this may look a lot like the DNA replication, and that as replication occurs, you could read along, and every single nucleosome, you just make sure that you add a methylated guy to each methylated site as you create the daughter strands, okay? But it turns out that it's pretty clear that those methyl marks require days to reestablish, and it's not something that happens instantaneously with the replication of the chromosome. And so there must be some other mechanism. Okay, and so what we've been exploring is first to look at work in Jerry Crabtree's lab, where what they've done is they look at undifferentiated cells, and they localize that chromoshadow domain of HP1 to a particular locus, and then they ask, as time progresses, what happens to the methyl marks around that locus where that chromoshadow domain happens. And in fact, near that locus, and this happens to be this is a particular locus. I'm going to show you the data on OCT4 in just a second. But there is a localized spreading around that region, which is to say that there is some communication. 2 minutes, thank you. There's some communication between the local chromatin architecture and the spreading. Okay. And so what we've done is we've endeavored to develop a basic kinetic model, which includes the local fluctuations of the chromatin fiber, the looping, as in the local looping of it, and the methylation and nucleosome turnover. And we have done this based on the local occupancy of the nucleosomes around the OCT4 nucleus. And we get predictions of the local spreading. And when you compare it to the experimental results, you see certain features like this asymmetry, which we believe is associated with this slight nucleosome-free region right by the transcription start site. And so in this regard, the local architecture plays some role in that conferral. And what we're doing now is we are moving to address how, when you actually have a more complicated segregation into heterochromatin, euchromatin, what role does that segregation play in the reestablishment of the methyl marks as on a generation-to-generation basis? Okay. So the very last thing, and I'm only going to take a minute to do this, is I want to say something about some experiments. Now, all of the work that we've done so far has taken essentially an equilibrium picture of the chromosome. All of the configurations that I've shown so far are based on equilibrium Monte Carlo, and that's probably a bad assumption. Okay, but the thing is that if we want to incorporate some kinetics and dynamics into this, we do have to acknowledge that the dynamics in a living cell is complicated. And one of the things that we're extremely interested in understanding is what role topoisomerases play in modulating the dynamics. Okay, and so the way that we've done this, and I have very little time, so I'm going to ask that you reserve questions to me after this talk. But we have developed. We've used dynamic light scattering, microreology, in order to describe the rheological properties of extremely small samples of biological fluids. We've done this for sputum samples for cystic fibrosis patients. We've done this for gut mucus. We've done this for a number of different cases. But for our purposes, I'm going to tell you about one which is kind of cool. Okay, and so here's what we did. If you look at kinetoplast mitochondrial DNA, it actually looks like this crazy mesh right here if you extract it, okay? And it actually is a bunch of plasmid DNA which is linked together into a bunch of catenated, like chainmail. You guys, some of you went to the medieval town, maybe you saw chainmail there. And this was dubbed an Olympic hydrogel by Dijon, okay? And so what we did was we synthesized an Olympic hydrogel out of plasmid DNA, okay? And then what we did was we measured the rheological properties for uncatenated solutions of DNA, catenated solutions of DNA, and then catenated solutions of DNA that has topoisomerase. And in this case, we're talking gyrase. And you're probably going to say, well, gyrase doesn't decatenate. But it turns out that we actually have a nicked segment of the DNA. And so gyrase, in this case, does decatenate. But the only point that I want to drive home, and since I'm out of time, is just to say that basically is that indeed we can make a Olympic hydrogel. We can take a dynamically topological network, and here this is the complex modulus versus the frequency. And for those of you who haven't seen a lot of this sort of data, it's just to say that where you have this region of frequency space where there's a plateau, that is an elastic plateau, and it behaves sort of like an elastic solid in that region. Okay? And so if you cut off the topoisomerase and it becomes permanently catenated, that plateau goes all the way to negative infinity. So we get that specific response. And the main point that I want to drive home, the last point, is just that. If you compare the rheological properties between an uncatenated solution of DNA, so they're able to move around each other, right? They're entangled, but they're able to move around each other, versus one which is catenated, but topoisomerase is able to function and able to delink, it lowers the modulus by at least a factor of like 6, and the relaxation time goes up by almost an order of magnitude. So in other words, it is better for something to have topoisomerase and be able to disentangle itself than it is for it to be entangled but not topologically linked. Okay, and I think this is an interesting system for us to explore issues of topology and relaxation in chromosomal DNA for an in vitro system which you can add the components systematically. So I'm going to stop here. I just want to thank all of my students who contributed in various ways to this talk. I'm extremely indebted to the Physics of Living Systems program for funding throughout the last number of years. And thank you. I'm happy to answer any questions.
We have time for a couple of questions while the next speaker brings their computer up and gets set up. Jose.
When you do your model for methylation, acetylation, and you say about flexibility of the DNA, do you use a uniform model or that's sequence dependent? Different places have a...
Yeah. So a couple of things. Yeah, that's an awesome question. And actually, that's at the heart of one of the things that we haven't submitted that paper yet. And the reason why is because, so I mentioned Bruno's work on there being heterogeneity in the linker, right? And I mentioned the fact that thanks. And I mentioned the fact that it will dramatically influence the fiber's properties, the physical properties of that nucleosome array. And so now we are indeed incorporating that heterogeneity and trying to work out what the statistics of that is. And it's actually really important for getting the shape of that methyl region. Okay. It is. So I don't have the data here, but yeah, that's
very curious to see what that is.
Yeah. Well, I'd love to show it to you. Absolutely. Yeah.
Hey, this is just a really specific point, but
Yeah.
it was a really fascinating talk. You showed this asymmetry in nucleosome occupancy in the vicinity of OCT4 binding.
Yeah.
And I wonder if you had any thoughts about how that kind of thing originates.
Okay. Yeah. So that's a very good question. It's also one that we have had considerable difficulty in exactly nailing down exactly the occupancy, because from a variety of different sources, the nucleosome-free region appears to be considerably variable near a transcription start site. And so at this stage, we're more being experimentally driven by where the occupancies are. We're not explicitly modeling what causes those occupancies. That being said, the region we fix, and then there's heterogeneity between where the nucleosomes are thereafter. And there's pretty good evidence that the basic occupancies, apart from some aspect which is sequence specific, tends to be sort of like a lattice type gas for the interactions. And so the basic distribution of the linker lengths, we base on that basic physical picture. But yeah, I mean, the more experimental evidence that we have that has to do with the sequence and specific to OCT4, the better. And so that's the best I can do at this point.
And there's
Yeah.
one more question here, and
Yeah.
then we'll call it.
I was wondering if in your model you allow also for bridging with HP1 neighboring nucleosomes. There has been
Yeah.
recent in vitro work where they have shown that actually HP1 rate can dimerize between two neighbors.
Yeah.
Is that allowed for?
Yeah,
in
yeah,
your-
that's
I
allowed.
see.
And to be clear, the way that the interactions are captured in the simulations is that they're captured through a field effect where every nucleosome that has HP1 interacts with the density of all nucleosomes around it, whether they are close genomically far or way far, right? So it's unbiased by anything like that. Does that
Yeah.
answer your question? Yeah.
All right, let's thank Andy.
Yeah.
Okay,